Okay, so as I was talking about the osmosis, so like how are we going to apply the same concept in different animals and plant cells? See, there is a thing called as osmoregulation. I'm just giving you a very small, you can say, um, a very small concept about the osmoregulation, not entire concept. Osmoregulation basically means the maintenance of water. So every organism does that, maintenance of water, and different organisms have different patterns. Suppose if I talk about the animals, animals basically do this same thing with the help of their kidneys. But how do plants do that? The plants do that by through their vacuoles. They do that through their vacuoles. Now, what is all this like? How do plants do that? That is not the topic of this particular chapter, but we can see the behavior of a plant cell with respect to the higher or lower water potential. Suppose that this is a plant cell, and this plant cell is just present inside the plant itself. We are not discussing a plant cell placed in the laboratory. We are just discussing a plant cell that is inside the plant. Maybe it's present in the leaf, or maybe it's present in the stem or root, whatever. So obviously you know about this. This is the vacuole. Suppose that there is high water potential outside. What is the meaning of high water potential outside? If there is in the tissue of the plant, there is higher water potential. Would this cell gain water or lose water?
If it has a high water potential on the outside,
Yes.
it would not be able to gain water as fast, right?
Why?
Because
What's the definition of osmosis, by the way?
um, osmosis, like
Do
the
Mhm.
how do you say it?
You can open the previous Jamboard and check for yourself.
Isn't it from a high concentration of water to low concentration of water?
Yes, but I asked you to use some different words. Just open the Jamboard and read the definition. The previous Jamboard. Are you saving the links for all of them?
Yeah. You said a random movement of molecules of gases or dissolved molecules in a liquid from an area of high concentration to an area of low concentration.
That is diffusion.
Oh yeah. Where's osmosis?
Almost on the
So
last sheet,
passive
maybe.
movement of water molecules from an area of high water potential to an area of low water potential. So if there's
Yes.
higher water potential on the outside,
Mhm.
then the
Yes.
inside would get more water because of osmosis.
So would it gain water or lose water? The cell.
Inside would gain water. So the cell would
Yes, the
gain.
cell would gain water. So simply putting it, whenever there would be there would be higher water potential outside, so it is going to gain the water. Now, when it would gain the water, there is very high chance that this all water would end up in the vacuole. It would end up in the vacuole and ultimately increasing the size of the vacuole to a great extent, pushing all the organelles on the peripheral portion, on the peripheral side, leading to a pressure from the inside that would be applied on the plasma membrane or cell wall. This pressure exerted by the water filled vacuole water filled vacuole is called as turgor pressure. It is called as turgor pressure. And the cell in this condition is supposed to be the turgid. It is in turgid condition. The name of the condition is turgid. This phenomena is called as turgidity, and the pressure is called as the turgor pressure. And why this turgor pressure is just there? Because the turgor pressure is a result of gaining of the water inside the vacuole due to higher water potential outside, and the vacuole has become larger. Suppose, did you understand this example?
Yes.
Suppose that the water potential, okay, from now I would be writing the water potential as WP. The water potential outside is equal to inside, is equal to inside. It means it is not going to gain no net movement of water. According to the principle, the water is supposed to be moving from higher to lower water potential, isn't it?
Yep.
But the water potential outside equals inside, meaning there should be no net movement of water. In this case, the plant want to take up water. What would it do? Now please listen very closely, very closely. It would take up actively take up some solutes inside. The plant would actively take up solutes inside. What do you think would happen when the plant would actively take up the solutes towards inside?
If it would actively take up some solute from the inside.
No, no, no. From the outside to inside. They are actively taking up solutes inside means that the solutes were outside. They took it inside. So what do you think would happen when they would take up the solutes?
Would the water potential increase inside?
Solute increases the water potential. Just go to the previous slides and see what I told you yesterday. Would the solute increase the water potential or decrease it?
Um,
Not on the previous Jamboard, on the previous slides.
I don't have the link for this one. I have the link for the previous one.
Okay, hold on. Just hold on. Only people with access. No, hold on. Now you can open it.
So, um, adding few solute molecules to chamber A solute would attach to water molecules, lower their kinetic energy. So that means it would lower the water potential, and
Yes.
so the cell would lose water.
Now, no, 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 no. Why would it lose water? If it is lowering the water potential, now focus on my words. When the cell would take up solutes from outside to inside, it would lead to lower water potential. It would lead to lower water potential. Ultimately, ultimately gaining the water, gaining the water. Now, what is the difference between the two examples? In this example, there was already higher water potential outside. In this one, in number one. In number two, the water potential was equal outside and inside. The plant needed water. So what did it do? It took some solutes to lower down its water potential so that the, there would be a water potential difference between outside and inside. Between outside and inside. When there would be a water potential difference between outside and inside, leading to lower water potential inside, what would it mean? The water would enter into the cells. So there are two ways the plant cell can gain water. One, if there is already higher water potential outside, the water would enter into the plant, stored into the water, and the cell would become turgid. Cell would become turgid. If the plant needs the water and there is no net difference between the water potential outside and inside, the plant would take up some solutes into it, lowering down its water potential so that it could gain more water. It could gain more water from outside. Are these clear, both the examples?
Yes.
I would suggest you, after the class, revise the whole topic of osmosis, because if you forget what is happening, what is the relation between the solute and water, you would be losing this concept. It
Okay.
basically depends upon where the solute is higher. Wherever the solute is higher, the water potential would be lower. You can simply create a relation. Solute is inversely proportional to the water potential. Higher the water potential, lower would be the amount of solute. But if there is higher the amount of solute, lower would be the water potential. So if the plant cell is taking up some solutes, it means it is lowering down its water potential. And if it is lowering down its water potential, it definitely means that the water is higher outside, so it would be taking up some. Is it clear now?
Yeah.
Next is very fascinating example. Suppose that this is a cell and this is a turgid cell. This is a turgid cell. And why can I say it's a turgid cell? Its vacuole is swollen, but it suddenly loses water. When it loses water, its cell wall structure would not change. Remember, cell wall structure would remain constant. The structure of the cell wall would not change, but the vacuole would become shorter. You can say this is the normal cell, or you can just say it's a flexed cell. Flexed mean it has lost a bit of water, but it's not dangerously, the water is not dangerously reduced, the amount of water. But during this procedure, how it would lose water? First of all, tell me how the plant would lose water. For that, what would you have to do to make the plant lose water? Would you have to increase the water potential inside or decrease the water potential inside?
Decrease outside.
You have to decrease outside. It means water potential would increase inside and it would be decreased outside. So plant would be losing water. Well done. Suppose that the cell becomes dehydrated, meaning that there is drastically lower water potential outside. Drastically low water potential outside. So it means it would become dehydrated. A dehydrated cell, you know that cytoplasm is 85% water. So a cell, if a cell loses water and it dehydrates, it means that it is going to lose not only the water from vacuole, but also from cytoplasm. So it is getting dehydrated. Once you would see that it is getting dehydrated, it would lose the water from cytoplasm and vacuole, and now vacuole would look like this. And the cytoplasm, after losing water, cytoplasm would also shrink. Now cytoplasm will look like this. This is the cytoplasm now. This is the cytoplasm now. It has been shrunken. It has been shrunken. This state of getting shrunk is called as plasmolysis. When the cell would lose so much water that it would not only lose the water. See, in this pattern, in this flexed cell, the water is lost only from vacuole, only from vacuole. It is not lost from the cytoplasm, but in this case, the cytoplasm and vacuole both are losing water, leading to shrinkage of vacuole and cytoplasm, and looking like the one I just drew. Got it?
Yeah.
So that is the situation of plasmolysis. Now you can say this is cell membrane, but you can notice one thing: the cell wall is still in its shape. Why cell wall has not been, you can say, shrunken?
Um, because it hasn't lost water.
Cell wall basically is, first thing, cell wall is rigid. If someone asks you why the cell wall does not shrink, it is very rigid, very hard, very rigid, very hard, and uh,
So, yeah.
does
So I
not
think
shrink.
we can form a finance transition.
So that is why the cell wall would keep its shape, but the rest of the cell would shrink. Is it clear?
Yeah.
Ongoing for.
Okay, now there are two things we need to discuss in the very next class, whenever we are going to take it. Number one is why turgidity is important. Why turgidity is important, or you can also say we can study the significance of turgidity. This would be our next topic. Secondly, we would have these two things are same. Secondly, we'd be studying the behavior of animal cell with respect to osmosis. So these two would be discussed in the very next class. All right.
Yep. Okay.
All right. Thank you very much.
Assalamu alaikum.
Salam.